Welcome to today's lecture on the applications of microarrays in molecular biology and genomics. Microarrays have revolutionized the field of molecular biology by allowing researchers to simultaneously analyze thousands of genes or genetic variations in a single experiment. Today, we will discuss 12 key applications of microarrays, from SNP genotyping to functional genomics. So let's dive into the details. SNP genotyping microarrays are powerful tools used to identify single nucleotide polymorphisms, SNPs, within a genome. These arrays contain millions of short DNA probes, each designed to detect a specific SNP. By hybridizing an individual's DNA sample onto the microarray, we can determine their genetic variations at specific loci. This information is crucial for understanding disease susceptibility, drug response, and population genetics. DNA methylation microarrays enable the measurement of DNA methylation patterns across the genome. Methylation is an epigenetic modification that plays a vital role in gene regulation. By hybridizing DNA samples onto methylation microarrays, researchers can determine the methylation status of CpG sites, allowing the identification of differentially methylated regions associated with diseases, development, and environmental factors. ChIP chip analysis combines chromatin immunoprecipitation, ChIP, with microarray technology to study protein-DNA interactions on a genome-wide scale. By immunoprecipitating a specific protein of interest and hybridizing the associated DNA fragments onto microarrays, researchers can identify the genomic regions where the protein binds. This technique is invaluable for understanding transcriptional regulation and chromatin structure. Comparative genomic hybridization arrays, also known as are used to detect DNA copy number variations, CNVs, and chromosomal aberrations. By labeling patient and reference DNA with different fluorescent dyes and hybridizing them onto microarrays, we can identify gains or losses of DNA segments associated with diseases such as cancer and developmental disorders. Microarray-based mRNA expression profiling allows the comprehensive analysis of microRNA expression levels. miRNAs are small non-coding RNAs that play a crucial role in post-transcriptional gene regulation. By hybridizing RNA samples onto mRNA microarrays, we can identify differentially expressed miRNAs and infer their potential roles in various biological processes and diseases. Microarrays can be used to measure allele-specific gene expression, which refers to the differential expression of alleles from heterozygous individuals. By hybridizing RNA samples onto allele-specific expression microarrays, we can determine the contribution of each allele to gene expression. This technique enables the detection of allele-specific expression patterns associated with genetic diseases and regulatory mechanisms. Alternative splicing is a crucial mechanism that generates multiple mRNA isoforms from a single gene. Microarrays can be utilized to probe alternative splicing patterns by measuring exon-level expression. By hybridizing RNA samples onto splice-sensitive microarrays, we can detect and quantify different isoforms, providing insights into gene regulation and potential disease-associated splicing events. Microarrays can be employed to characterize induced pluripotent stem cells, IPSCs, by examining their gene expression profiles. IPSCs are generated by reprogramming adult cells to exhibit embryonic stem cell-like properties. By comparing the gene expression profiles of IPSCs and their parental cells using microarrays, we can identify genetic signatures associated with pluripotency and determine the efficiency and quality of the reprogramming process. Whole transcriptome microarrays enable the simultaneous profiling of all transcribed mRNAs in a cell or tissue. By hybridizing labeled cDNA onto microarrays, researchers can capture the entire transcriptome and assess gene expression levels. This comprehensive analysis provides valuable insights into biological processes, developmental stages, and disease mechanisms. Microarrays have greatly facilitated genome-wide association studies, GWAS, by allowing the efficient screening of hundreds of thousands to millions of genetic markers. By genotyping individuals using SNP microarrays, researchers can identify genetic variants associated with diseases or traits. GWAS have been instrumental in understanding the genetic basis of complex diseases and individual variability in drug response. Dosage compensation refers to the equalization of gene expression levels between males and females in species with sex chromosomes. Microarrays have been utilized to investigate dosage compensation mechanisms by comparing gene expression levels across the sex chromosomes. This approach allows the identification of genes that escape dosage compensation and provides insights into sex chromosome evolution and gene regulation. Microarray-based gene knockdown experiments enable the study of gene function on a global scale. By transfecting cells with small interfering RNAs, siRNAs, targeting specific genes and assessing the resulting gene expression changes using microarrays, researchers can identify genes involved in various biological processes. This approach is invaluable for elucidating gene networks, identifying therapeutic targets, and understanding disease mechanisms. Microarrays are essential tools in molecular biology and genomics, enabling researchers to study genetic variations, gene expression, and more. They have greatly advanced our understanding of complex biological processes and will continue to shape the future of personalized medicine.